Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really fascinating guest involved in science and technology that's creating a better tomorrow for everyone. Today, we have the honor of being joined by Dr. Benjamin Oakes, who is co-founder, president, and chief executive officer of Scribe Therapeutics, a molecular engineering company focused on creating the best-in-class CRISPR-based therapies that ultimately permanently treat underlying causes of disease. Previously, as an innovative Genomics Institute entrepreneurial fellow, Dr. Oakes focused on a really holistic engineering of genome editing technologies to build these novel genome editing molecules. Dr. Oakes contributed to dozens of publications and patent applications across the areas of synthetic biology, molecular engineering, CRISPR, and zinc finger-based genetic modification. He received his Ph.D. in molecular and cellular biology from University of California, Berkeley. They're working in both the Doudna lab and the Savage lab, developing these CRISPR-Cas9 molecules with enhanced characteristics. And various projects resulted in these really distinct versions of synthetic sensing systems that will only modify nucleic acids with external stimuli, which have both research and pragmatic applied technology applications. Prior to his PhD, sort of in the before CRISPR days, Dr. Oakes received his bachelor's in philosophy and neurobiology and worked as a researcher in so-called unbiased combinatorial evolution methods to build literally tens of thousands of different genomic editing tools. A lot of really interesting themes to get into today. Very excited to have him with us. Dr. Benjamin Oakes, thanks you so much for taking time to come on the show.
Thank you. It's a pleasure to be here. I really appreciate it.
Yeah, it's great having you. A lot of really cool topics to get into, but where I'd love to start off is sort of, in the before CRISPR days, and here is a little bit about your early journey. I noticed in your publication, sort of before you did your dissertation, you were involved in everything from optogenetics to synthetic gene expression and yeast to zinc fingers, as we're mentioning. Talk a little bit about the days prior to CRISPR and a little bit about your background, if you would, Ben.
Yeah, no, it's fun to actually think about where synthetic biology was over a decade ago and what was really exciting and cutting edge. So actually my background starts out in undergrad. I was a double major in neurobiology and philosophy, which is like kind of weird, right? But that was when I really thought that ultimately I was going to go into the field of medicine, likely try to pursue something along the lines of an MD, PhD, because I was really interested in trying to get at the root causes of disease, right? Figure out how to build drugs. And it was only after kind of working in hospitals for a little bit during college that I realized that, quite frankly, we didn't have the tools to do that. And that's what really led me down this route of molecular engineering, protein engineering, and building really what was at the time the only reliable method for targeting the genome, which was really these zinc finger nucleases. So I was really fortunate enough to join a lab right out of undergrad as a technician, whose sole really meaning for being in life was to try to build a library of zinc finger nucleases that could be used by anyone. And back then, that was pretty difficult. Sangamo was really the only game in town for any academic, even, and of course for any therapeutic. And they were selling zinc fingers like 10 to 15 grand on pop through Sigma, and you could buy them if you wanted to edit a specific site in the human genome or any genome for that matter. But they wouldn't guarantee that they would work, and it was kind of a crapshoot, if you will. And so the goal of the lab that I joined was really to run tens of thousands of different selections to build a library of enough data to understand how we could provide essentially to anyone, to any academic, anywhere, the ability to design a zinc finger that could target the site that you wanted to. And I think what's so beautiful about that work in the early days is that not only was it incredibly valuable to really learn how to understand bacterial selections and what it means to engineer or to do directed evolution in a way that's meaningful. There's lots of really interesting and valuable nuance that comes only from, when you do your 1,000th selection on something. But also the really interesting thing was that while we were, of course, focused on trying to build zinc finger nucleases back then, with the goal of enabling genome editing in an organism, zinc fingers themselves and the ability to design a targetable zinc finger became incredibly valuable in many other areas, in these other areas, like you mentioned, like optogenetics, in these areas like building synthetic transcription factors in yeast. And some of my most fruitful projects actually became those where I was applying my expertise as a zinc finger engineer at the interface with, another academic, for example, Scott McIsaacs, who was working in the Botstein lab at that time, to essentially take those synthetic zinc fingers and build transcription factors to perturb what ultimately I believe they did, which was every transcriptional network in yeast. And it's kind of, I think, a lesson of when you build or are working on a technology that's so fundamental, like the ability to target the genome is so fundamental to biology, that there will be the opportunities are ever expanding. The more you grasp them, the more they become, and even beyond what I think you would have, what I thought when I was originally getting into it, which was we're going to be building genome editing proteins. And I think that goes doubly so now for CRISPR, and that's of course why I was so interested in not only staying in the field but expanding the uses of it.
Mm-hmm. Mm-hmm. And then seminal Doudna Charpentier paper is published in 2012. You start your PhD 2013 and published 2017.
Well, so seminal work is published in 2012, and I'm engineering zinc fingers, and I see it the day it comes out, and I turned to my boss at the time and said, "You'll never believe this, but like there's a programmable bacterial immune system, and it specifies DNA with an RNA in one-to-one fashion." Now, zinc fingers are basically 1 to 3. You have one protein module specifies to 3, and that's like complicated. There's a lot of complicated there's a lot of complex interactions that go on trying to understand how you can find a zinc finger binding interface, which is actually 6 or 7 amino acids to recognize 3 base pairs of DNA. And what's interesting is talons had come along, where these tail domains that were actually a little bit more simple. It was like 2 amino acids roughly, of course, in a very large backbone scaffold that specified 1 nucleotide. And that, of course, was deemed vastly superior to zinc fingers, but in the end was actually much more difficult. But then that paper gets published, and I still remember like sitting in this little cubicle off the lab, and just kind of being floored, and like going back to my boss Marcus Noyes was in the lab, in like the back of the gel room, and going back to him and saying, "You're never going to believe this, but this thing looks like the simple solution, like the obvious solution. And if it works, what we're doing is kind of," I don't think I would have said at that point obsolete, but I was like, but if it works, probably just like left the pregnant pause, right? and then walked out. Um, and that was the moment in which I decided, okay, so I'm going to go to grad school out at Berkeley with Jennifer and see if we can really um, continue to expand the use case of these things, because it was so clear to me, at least, someone who had been like toiling in the field of genetic medicine, genetic engineering, building genome editing proteins, how transformative this would be. That even if it wasn't to others at the time, like, you saw your life flash before your eyes and was like, that's where you have to go. Yeah.
You join the Doudna lab, and up until 2017, when you publish Engineering CRISPR-Cas9 Systems to Expand Functionality. And in your dissertation sort of lays, I mean, it's an amazing, body of work that sort of lays the basis for what Scribe becomes. So really three major parts, and I'll let you walk through this. One, obviously the RNA guiding RNA binding component. Two, sort of this deep mutational evolution technology, which you've been working on. And then the third part, this controllability, where you use this ligand binding domain of estrogen receptor, modulate it with tamoxifen, a topic we talked about on some of our breast cancer related shows. But talk a little bit about your dissertation here, because I think it's important in ultimately what Scribe becomes. So talk a little about this, if you would.
Yeah, I think it's interesting. The way I think about my dissertation in the labs of both Jennifer Doudna as well as Dave Savage is, I, of course, was coming in with this background of protein engineering. Really, first and foremost, how do we build libraries of tens of thousands, tens of millions, actually even billions of different molecules, and select them to identify not just one, but to identify all of the possible ways. For example, you could bind a 3-base pair unit. And actually, I look back and now realize that sort of approach was somewhat unique even at the time, right? It wasn't about selecting for a single thing that did the job you wanted. It was about capturing all of the information that occurs within a bacterial selection, and using that information to better inform how to engineer the protein kind of writ large. And that is a thesis that I had spoken about with another professor at UC Berkeley, Dave Savage, who I was co-mentored by. And Dave's lab was really focused on kind of doing these sorts of large, comprehensive mutational, and not even mutational, really topological screens, which was really interesting to me. And with the targeting problem solved, which is how do I target somewhere in the genome, right? If you solve that with an RNA, all of a sudden there's this really interesting, fertile ground of protein and molecular engineering that becomes now available. It becomes not about how do I target DNA anymore. It's how do I make a molecule that's more effective, that's more potent, that's safer, that is more useful. And my PhD really focused on, I think, these two things. I took a number of methods that were being developed in the Savage lab, coupled them with with a lot of my deep expertise in engineering DNA binding domains, and really brought that to Jennifer's lab, which was a biochemical lab at the time, right? And set up what was the first, as far as I know, really comprehensive screening systems to look at every possible change you can make in a CRISPR molecule, and start to understand what that meant. Quite frankly, in the beginning, it wasn't even, okay, we're going to build an allosteric Cas9. It was, if I can insert one domain into Cas9, where can I? How can I essentially do protein on protein accretion? How can I put two proteins together? And what does that mean? And we realized that, oh, wait, we can insert and we can insert domains into Cas9 in many different sites, hundreds of different sites. And then the question becomes, okay, if we can do that, which was surprising in and of itself, what does that mean? And quite frankly, that was the first comprehensive map of how you can essentially inlay domains into Cas9. People are now using this around the world for inlaying things like base editors, inlaying things like prime editors, for making better versions of CRISPR molecules. But we kind of set out with this original idea of, okay, if you can inlay a domain, maybe we can get one domain to kind of control the activity of Cas9 in another way. And that's where we brought in the estrogen receptor ligand binding domain, which was an interesting domain because it's known for having actually quite a large conformational change upon ligand binding. And it's known for actually having different ligands that make it that change how that conformation looks. So it's kind of this perfect test case. And long story short, we were able to demonstrate that we can control essentially the activity of Cas9 by using the conformational changes of the estrogen receptor ligand binding domain in an allosteric way versus some of the other ways that folks have used that in the past. My PhD then extended beyond that to look at, well, if we can insert domains into Cas9 and make them more controllable, what else can we do to this protein? We actually did something called circular permutation, where this is quite esoteric, but you can essentially attach the beginning and the end of the protein together and open up new beginnings and ends. And the protein still folds in the same way and behaves in the same way. And that's actually very useful because a lot of the use cases for Cas9 are actually fusions to the beginning and the end of the protein. So positioning that around the protein in different ways allows you to kind of access entirely different activities that would have been less effective or hindered in another way. And so you kind of put this all together, and what we realized was that CRISPR molecules are really interesting, highly evolved tools in bacteria. But when we get our hands on them, whether for synthetic biology purposes or for tool purposes, there's really almost no end to the engineering and the changes that we can make. That's really what my PhD, I think, taught not only myself, but a lot of people. We're doing crazy things. We're taking entire proteins and sticking it into a CRISPR molecule, and it's still able to edit the genome. And this really, I think, informed a lot of the philosophy behind Scribe, as well as, of course, all of that work underpins a lot of our understanding of how we evolve proteins, right? A lot of the methodologies for protein evolution, and really, I should say molecular evolution, because we evolved the RNAs as well, was really underpinned by all of that work in terms of understanding just how far these bacterial immune systems could be pushed.
Yeah.
Yep. And so then, when I graduated in 2017 and was fortunate enough to be awarded this entrepreneurial fellowship, the question became not a question of just how far can we push it, right? The kind of the very, I would say, exciting grad school question of just how many cool synthetic biology tools can we make, right? How many different types of switches can I make? In the end, we made a Cas9 that turns on in response to viral infection. We got pretty deep down the rabbit hole. But instead, can we, back it up and take a broader look at the whole field of genetic medicine, where that was in 2016, really. And what we realized is that almost everyone using CRISPR technology had essentially just co-opted it from nature, co-opted it from what was being seen in academic labs, and started to apply it as quickly as possible. And that meant that the majority of the companies, even today, who have seen really great success with CRISPR technology, have done so with a molecule that's really just adjacent to what a bacterial immune system evolved for its own purposes. And quite frankly, that's not, when I'm feeling ungenerous, I kind of, I say that's somewhat akin to doing surgery with a stick, right? It's not what humans do best, which is we take the raw material that nature gives us. Quite frankly, we melt that down entirely. We reshape it to our will such that we build tools that are fit for purpose. Well, we have not done that with CRISPR. And that really seemed like that was the gap that Scribe needed to fill.
Again, you just were pointing out again per the work you did in your dissertation, issues with regard to the evolution of its activity, its safety, and the ability to change sort of the therapeutic payload, let's say. But then to further, I mean, you went a lot further, and I guess this gets into sort of the XE technology and some of what we're talking about per Scribe, but other components to this, I guess, this four-legged stool, namely these tropism factors that allow, the protein to go certain, or the CRISPR to go certain places, and then enhance its delivery. And then some of these other, and I'll let you talk about the polyproteins and everything else, sort of that say, hey, immune system, don't look over here. Look at other stuff. We need to bypass existing immunity challenges. These four aspects, delivery, activity, safety, positioning, come together into what the XE platform is. Can you talk about that as well?
Yeah, I guess so. From this foundation, or this somewhat um, overly simple realization that we have not engineered these natural bacterial immune systems to be the genome editing scalpels we need. We highlighted just again a couple of really important, critically important therapeutic characteristics that we would need to really transform what we believe the future of genetic medicine should be, which is genetic medicine for everyone, right? How can we, as I mentioned earlier, my goal early on was to try to figure out how to build drugs that could be utilized to get at the root causes of disease for everyone. And in the end, it's, when we think about CRISPR and genome editing right now, it is somewhat relegated in many people's minds just to rare disease. But if we want to be able to move beyond rare disease, if we want to be able to treat anyone and everyone for, every condition that we're all going to get as we age, we started to ask these questions of, what would those technologies really look like? What would they require? And to your point, the simple answer was like, well, shit, we need something that is perfectly specific. It can't edit off target because, if you have off target risk and the risk benefit is there in a patient population of 1,000 people where the disease is devastating, that's fine. But if you want to treat 1 million people, 10 million people, or 100 million people, which you certainly do for many diseases, you can't take a 1% or a 0.1% off target risk. And so first, let's build things that are, as close to ideally specific as possible. Second, of course, is you need something that's as potent as possible, because delivery into the human body is always limited. It's always going to be limited, whether that's via viral vector or via lipid nanoparticle, which are the two core ways that most biological components are delivered into the human. And so potency becomes critical because your delivery systems are limiting. And so we have these two kind of aspects, potency and specificity. And then the next one, to your point of delivery, is, I think, even more important. So there are the current limitations for CRISPR-Cas9 molecules. Many of them were significantly too large to utilize a number of viral delivery systems that we currently have. And so we wanted to ensure not only that we had a molecule that was small enough to take advantage of the viral delivery systems that worked, but also one that we could actually engineer the delivery components themselves, such that we weren't just engineering the CRISPR molecule, but we were engineering the entire package we were trying to deliver into the body for, again, improved potency, improved specificity and safety, as well as a number of other aspects. So, even just one of those things would be challenging. And to kind of take on a multitude of them all at once kind of became our mission of how do we build a genome editing platform that has specificity that's as exquisite as possible, and that has potency that can rival what we currently have, because we know that works, right? We have approved drugs with Cas9 based molecules now, but hopefully exceeds it. And that is really what we've been focused on doing for the X editing platform, or what we call the XE platform. We evolved this thing starting out from an entirely new molecule called Cas-X, which was discovered in Jennifer's lab. That molecule is smaller and significantly different than Cas9. It's like, less than 5% identical. So it's a really interesting and unique starting point. But it also was one that I think had a number of really interesting inherent advantages in terms of how it cut DNA, and its overall specificity to start with. The core challenge there was that molecule, the Cas6 molecule discovered in Jennifer's lab, basically barely edits the human genome. Like, on a target that is on most targets, you're getting between like 0.5 and 1% genome editing. Now, of course, that's nowhere near enough to be therapeutic. And so what we did is we undertook, again, the comprehensive and iterative engineering approach that we had brought to bear, that I had brought to bear during my PhD, but now really, I'll say initially on these two axes, activity and specificity, right? And we did essentially a dual axis set of selections and iterative evolution that has allowed us to essentially improve activity over 100-fold and create a molecule that does not cut off target even when used at 10x a saturating dose now in non-human primates. And that is really the core molecule that we call the X editor.
Mentioned, Ben, saturating there, and I'd love if we could talk a little bit about that, because you showcased several aspects of the XE technology at the American Society of Gene and Cell Therapy meeting a couple of months ago, and one of them was this saturating of hepatocytes and interesting non-human primate model. Talk a little bit about, because a lot of these we think a lot about the genetic disorders, and we'll get to the non-genetic stuff in a little bit, but we think about the blood disorders, but clearly, and when again looking at some of the stuff the Scribe's going after in terms of multi-system disorders, neurological diseases, muscle, and so forth, what is required in the case of an organ when we have a genetic disorder in terms of this principle of saturation? When we talk to folks like Tony Atala down at Wake Forest, who's active in the organ bioprinting space, he always points out, well, if I have a damage to my heart, whatever, my heart still works for a while until it gets really bad. What are we looking at when we talk about saturation and some of these genetic disorders in major organ systems, of how far we have to go with the gene editing process?
Yeah, it's an interesting question. I think what is interesting is actually even with some of the genetic disorders, you may not, and it really depends. And this is the hard part about genetic medicine, is that every disease is its own disease, right? So there's not a one size fits all answer for this. But what I would say is that while some diseases you may need to only fix 10% of cells, to your point earlier, right? Like that's sufficient to get you enzyme replacement in some diseases. From the perspective of how do you build a genome editing tool that can be used where you want it to, you need to achieve essentially editing of every cell that you deliver to, right? Because again, we're delivery limited. And therefore, I think what we demonstrated earlier this year at ASGCT is that outside of Cas9 based data, Scribe has built the first, and I believe to this moment, only technology that's non-Cas9 based that can achieve editing of essentially every hepatocyte in a non-human primate liver, which is pretty astonishing, right? Like that's it seems like that's almost a bare minimum bar that we should have to get over for genome editing. And the original molecule that Jennifer discovered, S-pyogenase Cas9, can do it. But to date, I have not seen data really suggesting that there's another molecule that can do that well, and certainly not another molecule that can do that well as the X editing platform can, while also achieving the specificity that the X editing platform can. And so what we can see is basically we're able to edit greater than 75% of cells in the whole non-human primate liver. That's about the percentage of hepatocytes that's expected to exist. And because LNPs are preferentially delivered to hepatocytes, I think the realization is that we're essentially editing almost every hepatocyte in the non-human primate liver. We've also seen that when we, for example, double the dose, we don't observe any more editing, again, indicating that we are editing where we're delivering. Now, you couple this fact, the ability to saturate genome editing in the non-human primate liver at therapeutically relevant doses, with the fact that when we actually do off-target studies for our molecule, for one of our first programs, for example, which is trying to build a drug that will lower LDL-C or bad cholesterol. When we actually look at the off targets of this program in primary human hepatocytes, which is where, of course, we have to look at them, the human genome, we observe that we see no off-target effects to date, even as we continue to increase the dose above this saturating dose, to kind of be super saturating or the non-physiologically relevant doses of almost 10 times the amount. Now, this is still early data. I would say that we're still, obviously, we want to make sure that we are as diligent as possible, seeking out every possible off target that there could be. But so far, we have not seen, I think for really the first time, again, a molecule, we have not seen any off targets. And so this is really interesting because it starts to raise this question of, what if you could do genome editing or you could do genetic medicine without the risk of an off target? What if we can build a molecule that was built not to have that characteristic? And what if we maybe already have? How does that change people's thinking on the risk benefit of genetic medicine? Given that anytime you ask anyone, the question is, well, I don't want to, I don't know that I want to think about the off target risk there. Well, what if we just remove that from off the table? It's kind of an interesting idea. It's a world we'll certainly get to. And it's one that I think Scribe may already be pushing us to today.
So, most definitely. No, it's you're clearly making quantum leaps here with some of these results. And again, we'll put a link to the ASGCT to press release and everything you were disclosing at that meeting. Thinking a little bit about the ex vivo implications, because you are active in hematopoietic disorders, cell therapies, and that that can range from anything from, hey, I want to CRISPR this specific monogenic disease, sickle cell anemia, whatever, to something like CAR T, where okay, there's a couple genes that we got to play around with here, to the other end of the spectrum, to areas like induced pluripotency, which you may be working with 4 Yamanaka factors or more, and so forth. Talk a little bit about some of the things that you're learning, seeing in sort of the ex vivo space, because clearly, your tool has a role there, but now you got to start packing on multiple genes to do multiple things. Take us into that direction as well, if you would.
Yeah, that's interesting. So the ex vivo space is actually not a space we're working in overly much.
Okay.
We have a partnership with Sanofi,
Right.
where we're applying our technologies there, but we have focused almost exclusively, we have focused exclusively internally on building drugs that can be brought into the human body. And that is because that's where we believe ultimately genetic medicine should be used. I think taking cells out of the body and giving them back is a really important step, maybe a stepping stone, if you will, to bringing all of these medicines in vivo. And it does allow you, as you point out, to do a lot more complicated manipulations. I think what's interesting about that is what I've seen in the field is that, as these manipulations, to your point of, ex vivo cells get more and more complicated, again, the technology needs to improve, right? We can do editing of 2 to 3, maybe 4 different sites all at once ex vivo. But if you really want to start to think about entirely reshaping a cell, you do need to start to look at, or you would be better off perhaps starting to look at things like epigenetic modulation rather than straight up genome editing, because that has the added benefit of not having risks like translocation between multiple genome editing sites. And so this is actually, it's a nice segue to, if I could, tie into the second technology that we built, which is an epigenetic silencing or an epigenetic modulation technology. So we call this thing the epigenetic long-term X repressor, which is like just a mouthful. So I'm going to call it, or so we do call it, the elixir for short. And the elixir, yeah, it's a good name. It's fine. It's fun. The elixir is a targeted epigenetic silencer that allows us to do some of the same things that you would want to do with an editor sometimes, but also an entirely different set of things, which really is targeted genome silencing or down regulation. Doesn't have to be all the way, in a way that is not permanently modifying the genome, which allows for, again, this really intriguing concept of being able to reactivate the gene if you want to, or having regulation, down regulation that lasts only for a certain period of time. And this is, I think, a really, again, intriguing way to think about the future of medicine, because every molecule, small molecule or antibody that currently exists that downregulates a target, which is the majority of them, right? It's quite a bit. You could ultimately imagine replacing with an epigenetic silencing technology that does the same thing in a targeted way, but could also be turned back on. So rather than taking an injection every week or every month or a pill every day, you could imagine a world, and I think we will see this world in the next couple decades, where you go into your doctor's office and you get an injection or an infusion, and then that modification is kind of taken care of. And if you need to, for whatever reason, turn it back off, that's also well within the remit of the medical professionals who are working with you. It provides like a really, again, a much more nuanced view of how we can treat biology, perhaps, and how we can kind of tip everyone towards the scales of a healthy, living a healthier life, compared to how we think about medicine today, which is really mostly symptoms based, mostly based on treating folks once they get really sick. And that's where I think is really interesting, is once we start building these tools that, again, are safe and effective enough, safe and potent enough, we can really start to envision an entirely different framework for how we're bringing medicine to bear.
Sort of the elixir outline quite elegant. Again, you're using your novel Cas to not just find the genome, but hey, we're going to put other things on here that either slash off that methyl group or do something with the histones. And yeah, I think
That's exactly right. Yeah.
the on the non-genetic front, as you were pointing out, because here, one of the hot areas that we've profiled a bit on the show has sort of been this epigenetic reprogramming, ways to do it in vivo that we could, again, if I have some scar tissue in my heart and I want to turn it into muscle tissue, that's more than one gene. As you were pointing out, we will look at the epigenome, and it seems like your tools are the ideal candidates for getting something like that done at that level of sophistication. So I was very excited just reading that part of the presentation.
Yeah, I think it's an interesting question of just how we get to that level of sophistication in vivo. I want to say I don't think we're there yet. I don't think we or anyone else is there yet. I think it's important to recognize that there's still a long way to go, but we're certainly marching in that direction. That's the hope.
I just want to ask again from the recent meeting. You also presented on an interesting model of retinitis pigmentosa, and again, we were talking about when we get back to the genetic diseases and sort of the orphans. Clearly, we got a lot of them out there, somewhere 8 to 10,000 of these diseases. Most of what we talked about in terms of addressing them pharmacologically has been sort of in the repurposing space, but clearly, what you're doing here. Again, it seems like it's going to be not just, hey, I can take care of this, but little tweaks along the way. We can move from retinitis pigmentosa to I'll pull genetic disease X out of the air, but it seems like a lot of plug and play stuff here, and that can happen rather rapidly to address quite a wide range of these diseases that are unmet and stretch the imagination in 2024.
Yeah, I think the retinitis, the retinitis pigmentosa, the ADRP story is one that's interesting, because it is one where we again are showcasing kind of the programmability or the engineerability of our platform, as well as the just extreme specificity. So we actually built a molecule, purpose built a molecule to demonstrate that we can target an entirely new site in the human genome that was actually untargetable by the natural Cas enzyme. I won't get too deep into that, but basically we re-engineered the PAM to make that happen, and the PAM is the only hard-coded region of a CRISPR molecule. that recognizes DNA. So in Cas9, it was GG, GG dinucleotide, and the original Cas6 was a TTC trinucleotide. And so we re-engineered that to demonstrate the ability to actually recognize a broader site. And then we actually, again, brought in a level of specificity that allows us to edit only the mutant allele while leaving the wild type allele alone, which is what's necessary for some of these autosomal dominant disorders. And I think it really represents a really promising use case for, of course, this technology, to your point, in being able to go from one disease to another quite rapidly once you create a molecule that actually has the potency and specificity that it needs to do so. So I think it's again, it was a really beautiful story around just how quickly and how effectively we can do this at Scribe from a molecular engineering perspective.
Really cool stuff. I want to ask about, last year, I hosted Brad on the show from IGI, and we talked sort of broadly about Innovative Genomics Institute and all things CRISPR. And other things outside of sort of the Scribe targets, whether we're talking agriculture, the microbiome, other things that we that are sort of on the drawing board currently. What else gets you excited about the potential use of these tools beyond Scribe's portfolio, but things that you envision 5-10 years out in terms of applications of a tool like this?
Yeah, I honestly I can't oversell the importance of being able to modulate the genome, right? Like, at some level, you want to be careful about that because people think that, okay, the genome's the code of life and that's all there is. But like, literally, I was reading a paper just a couple weeks ago, or maybe even just a week ago or so, that was talking about like inheritance that's based off of proteins, like amyloid-like proteins, and which is like a completely new idea. We've never, we're always going to find new stuff in science, which is, I think why I love it, right? It's always exciting. But the genome does provide a really large and fundamental portion of the code of life. And up until now, really the past decade, we have not had the ability to manipulate it and break it, quite frankly. And like, if you can't take apart the clock, you can't understand the clock. And so I think that one of the things that gets me the most excited about these technologies is, quite frankly, just the basic research that we are going to be able to do to better understand how the genome itself works. And it doesn't get the credit that, for example, editing in plants does, or editing in microbes does. Like, the actual, we're using this tool to change something. It doesn't get that credit, but like, it deserves more than it, because you can't change what you don't understand, right? And so fundamentally, I'm most excited about being able to take these technologies, whether it's basic genome editing or the precise technologies that allow us to change single nucleotides to other nucleotides, and explore what happens if you make every change in the human genome, or, quite frankly, obviously not the human genome. Can't really start there unless you're in cells, but in any genome. And if we change this nucleotide to that nucleotide, it's not going to matter 90% of the time. But every now and then, we're going to realize that, okay, this SNP, this single nucleotide polymorphism, actually underpins a huge and dramatic change in health or effectiveness of some sort of process within the body. And when you start to think about coupling that with, of course, the sequence, the experiment we've already run as humans, which is, there's 8 billion of us or so, probably going on 9. If you could get just a small portion of that sequenced and start to unpack what those changes mean, and then use genome editing to study them, in the lab, we could really start to understand how to master our own genome in a way that allows us to all lead dramatically healthier lives. But that is much more difficult and much longer term, I think, than we give it credit for, perhaps. But I also think perhaps much more important than we give it credit for as well.
It's going to be an exciting, The mission of your company is engineering the future of genetic medicine, and it clearly it's going to be an exciting future to watch how these tools evolve and what comes next. But I'm rooting you on. I'm excited about it all, Ben. We talked a lot about ASGCT, but anything else coming up that we should know about sort of for the next sort of this following quarter of the year? Other conferences, other initiatives that are public facing as we head into 2025 while we have
Yeah.
you today, please.
Yeah, so I mean, again, we'll continue to present. We just talked a little bit about we had a presentation at Cold Spring Harbor,
Mhm.
which is another conference about some of our epigenetic silencing technologies. That was exciting. We are, as a company, internally focused on addressing, again, the largest diseases that we think we can have a really strong impact on, and that for us is cardiometabolic disease. And so we're excited to actually be presenting some of our work coming up at AHA as well. So I would keep an eye at that on that and some of our developments, not only around LDL-C lowering, but perhaps even beyond that.
Again, I'm excited for you. Wishing you and the team the best with all this as you move the portfolio forward. Again, for everybody that's going to be listening to this episode of our show across the podcast networks or watching on the YouTube channel, you've been spending time with Dr. Benjamin Oakes, co-founder, president, chief executive officer, Scribe Therapeutics, engineering the future of genetic medicine for all of us. Ben, I really want to thank you for taking the time out of your schedule to talk to us for a little while, walk us through what you're doing. Thank you for everything you do. And as we like to say on our show, again, thanks for helping to create a better tomorrow for all eight, close to 9 billion of us with these technologies that you're developing. Really an awesome story.
Oh, thank you so much. I really appreciate it.
Great having you.